Hello everyone, my name is Harry. I'm currently a master's student in Department of Biomedical Engineering of Chung Yuan Christain University. And currently I'm working in Center of Aquatic Toxicology and Pharmacology. And my advisor is Professor Chung-Der Hsiao. And today I present my work. My title is Investigating the Skin Wound Healing Potential of Biodegradable Polymers, PDLLA and PLLA in Zebrafish. For easier understanding, I will separate my speech into four parts. First is the introduction, second is material and method, third one is result, and the final one is conclusion. And recently, my research have been published on biomedicine and pharmacotherapy with the impact factor 7.5. And this is my experimental overview, including the wound healing assay. This is the most important assay in my research. And molecular investigation. So we detect the mechanism of wound healing using the RNA through RNA isolation and qRT-PCR. And we summarize some of the healing outcome by the skin impression. For the introduction, we all know skin is very important for us. It's the largest organ, can protect our body from invader from outside and maintain our body as the balance stage. So when we get the wound, we need the wound healing process, and this is a very complex process, and we require to be fast, coordinated repair. And this study using the zebrafish as an animal model to study the wound healing. So why the zebrafish? Because the zebrafish has some advantage, like and the low cost, easy to handle, and similar healing process to mammals. So the method we use to inducing the wound on the zebrafish for the wound healing study is using our lab developed laser-based ablation for precise wound induction. And after we get the wound model, we access biomaterial property in wound healing using the PDLLA and PLLA. So these biomaterial are safe, biodegradable, and aid regeneration. So our aim is using the precise low-cost method to assess the biomaterial therapy for wound healing application. So there's some impact of this study. First is to provide insight about the wound healing process under treatment, specifically biomaterial treatment. And we provide a zebrafish platform. It's a fast ethical model for studying the healing and testing new therapy. So why using the zebrafish for wound healing? So first, zebrafish is a small, around 2 to 3 centimeters fish that's very famous for medical research. So its skin is healing very fast. So it's fast skin regeneration, and it has transparent body for life imaging. And most important thing is genetic similarity to humans, around 70%, and low cost and easy to maintain. And they already have a well-characterized immune system. So the study objective is develop a standardized zebrafish platform to access biomaterial, which is in this case is PDLLA and PLLA, effect to the wound healing. So through that we evaluate wound healing and morphological assay and behavior alteration to prove that the biocompatibility of the biomaterial. And also we investigate the wound healing mechanism in molecular level. So I make a table for the comparison of wound healing models. So I compare zebrafish to 3 other traditional models, and like the mouse, the rabbit, and the pig. And you can see here zebrafish have some huge advantage compared to traditional model, like the skin regeneration, the imaging capability, the genetic tool available, cost maintained, throughput, ethical regulation. So using the zebrafish, you can have a high n number sample for more reliable data compared to mouse, rabbit, and pig. And also, it can save your time because the zebrafish healing faster than the bigger traditional model, and also the cost of maintaining is very low. So using the BDLLA and BLLA as a healing agent, as a biomaterial. So BDLLA and BLLA are biodegradable, biocompatible polymer using in medical and cosmetic application is already well known. and is famous for the remote collagen production, cell growth, and tissue regeneration. So offer controlled degradation and better mechanical properties than natural polymer. This is a synthetic polymer, and this is very commonly used in suture and dermal filler and tissue engineering. And narrow in skin wound healing remain underexplored. So that's why I'm selecting the PDLLA and PLLA for this study. And this study evaluated their healing effect and mechanism in zebrafish model. And you can see this is the structure of PDLLA and PLLA. So for the material and method, so for the zebrafish study, very important for us to maintain the zebrafish under lab condition. So here we get the idea from the reference papers. So we maintain the zebrafish BAT strain, collect from the best store or Academia Sinica, and keep them in 10 liters plastic tank at 28 degrees and fit twice with Artemia or dry food. And 50% of water replaced quickly with fresh reversed osmosis filter water, and the pH will around 7 to 7.5. And very important to maintain under 10-hour dark and 14-hour light cycle. And we also monitor the health condition of the fish. So if the fish show no sign of infection and healthy, we can use them for experimental. And this ethic already approved by Chung Yuan Christain University. And next, because using the animal model, we require to for the anesthesia protocol. So for the zebrafish anesthesia, I'm using the 0.1 ms22 for the unconscious fish, and this is the safe and non-lethal concentration. So simply just prepare ms222 solution, and then I will place the zebrafish into the solution for around 10 to 30 seconds, and I'll observe if they don't have any sign of moving. I will collect for my experiment like the ablation for the oral administration, and after that I will move the fish to recovery tanks for 30 minutes until fully recovered. So this is the overview for the skin ablation protocol by using the laser engraving machine developed by our lab. So this is the figure of the laser machine. So A is the total laser machine, and the E we can see here this control. This is the control panel for in the software for us to control the power and the distance and the speed of the laser machine. And for this study, I already know the optimal condition for the skin wound healing by the previous study we published in our lab. So just based on that optimal condition, I doing my experiment. And this is my most important assay in my study called the wound healing assay. So this is my experimental timeline, and based on the timeline, you can see I would change the water every 2 days, and I would do the oral gavage 2 times a week to make sure that the biomaterial effect, and we can get the highest the wound healing outcome. And for this the wound observation for the calculation, I will collect the image every 5 days and using the image age to calculate the wound size, and through Prism I can generate the graph and data analysis, so finally I can get the wound healing curve. And this is for molecular investigation of the wound healing mechanism. So through tissue dissection and QRT-PCR for gene expression. So first I will collect the skin, the wounded skin of the zebrafish after 10 days treatment. And through this protocol, I finally will collect the RNA, and using the RNA for QRT-PCR assay, and then I can collect the gene expression profile. So for the biocompatibility of the biomaterial, I will do 2 assays. First is the embryonic development. So I will export the biomaterial 24 hours after the fish laid the eggs. So basically I have 4 groups: the control, the BDLLA treatment, and the combine between 2 polymer. And I will observe and collect the image every 24 hours. And I'm using the concentrations is 1 ppm because it's equal to 1 milligram per kilogram dose for the augmentation in the adult zebrafish. And I also use image A for morphology alteration calculation like fish length. So the second assay for the biocompatibility of a biomaterial will be the 3D locomotion test for the behavior analysis. So zebrafish is very sensitive to chemical, and we can observe that through the behavior alteration. So we're using the 3D locomotion. So 3D locomotion protocol already been published by our lab too, and I'm using that protocol to collect the video. And the video will track the zebrafish swimming pattern in 3D, and through UMA tracker I can track and then get the data. And using another app developed by our lab called the F3LA for the data analysis, and we can collect the graph to understand the behavior alteration by inducing the biomaterial to the zebrafish. And third part is the result. And for the result, I will start with the image analysis of the wound healing in zebrafish model under different condition. So we have a control and different treatment like the PDLLA and PLLA. And after I know that PDLLA and PLLA have the effect to push the wound healing process, I will combine them to study the synergistic effect with 3 different ratio, the 1:1, 1:2, and 2:1. And after I select, I already know the optimal combination for the wound healing boosting. I will use that to study the dose depending effect. And first, I'm using the 1 milligram per kilogram dose for the screening step. And based on that, I will select a lower dose and a higher dose for us to more fully understand about the dose depending effect of the biomaterial treatment. And additionally, I also compare with to the well-known wound healing agents like the antioxidant. Here specifically is astaxanthin and vitamin C. So on easy observation, we can see that using the biomaterial with the same dose and the same administration route, like specifically is the oral biomaterial have higher boosting property in wound healing compared to astaxanthin and vitamin C. So this is the most important data for this research called the wound healing curve. So the wound healing curve will tell us specifically the wound healing property of the biomaterial through numbers data. So this study evaluated this polymer individually and in combination to assess their effect on the wound healing. So we focusing on early, mid, and late stage of the wound healing, and the process is 50 days. So from the result, both PDLLA and PLLA accelerate the healing compared to the control. For example, PDLLA achieving 25% closer only took 9.6 days compared to 17.4 days of the control, and the PLLA is 11.9 days, still faster than the control. And also the 2:1 PDLLA and PLLA combination was the most effective, reaching 50% closer in only 15.4 days and 75% in only 23.5 days. This is outperforming every individual treatment and other ratio. So after that, we know that 2.1 ratio is the most effective. We will use that to study the dose depending effect. So for the first depending effect, 0.1 milligram per kilogram dose led to the moderate improvement, and 1 milligram per kilogram was the optimal dose. And why the 1.5 milligram per kilogram was the higher dose? It's so effect in the early healing, but later showing slower than the 1 milligram per kilogram. This suggesting the high dose may hinder long-term healing. And interestingly, the tested antioxidant, which were the astaxanthin and vitamin C, proved to improve the healing over the control group, but were less effective than the polymer treatment. So astaxanthin outperforming vitamin C in the early stage, but fell short compared to the polymers mixture. So for the conclusion, overall, we can see that 2:1 PDLLA and PLLA combination at 1 milligram per kilogram provide the fastest and the most sustainable healing. So this demonstrates the prospect of the optimizing clinical wound healing protocol. And this is the support data for the wound healing curve. This is the survival rate of the treatment. So we can see here the zebrafish under a lot of bad condition like the stress from the laser ablation and the oral gavage. But even though with that stress and with the treatment from the biomaterial, the survival rate is still acceptable. Every treatment have the survival rate higher than 80%. So this indicate a good biocompatibility of the biomaterial across doses and combination. So mentioned about the biocompatibility of the BDLA and PLLA on the zebrafish through the embryonic development. So through figure 8, you can see here the 24-hour HBS to 120 HBS. The zebrafish developed with no alteration in morphology. And through calculation the body length, there is even though it's a slightly differentiated between the group, but it's not significantly worth to mention. And also the survival rate also support our hypothesis that biomaterial BDLA and PLLA is very safe and have the high biocompatibility to using in the wound healing study. So I will show you example because if the chemical have some effect to the pathology for in the embryonic development, it will show the image look like this. This is from the reference papers. The zebrafish under another chemical and have some bad effect to the morphology. So in figure C and D, you can see that the morphology is significantly altered by the chemical. And this is the result for the 3D locomotion assay. So there is some conclusion while you can observe through the figure. And the note is there no maturity over 14 days and all the treatment are safe and non-toxic, because in this study I didn't do the laser ablation, so the fish only under condition of the oral gavage, and with the oral gavage only there's no maturity or over 14 days of the observation, so the treatment are safe and non-toxic. So I have some conclusion I extract from the figure is BDLA is altered locomotion in like speed, freezing, and angular physiology, but all of them is not significantly different. And PDLLA and PLLA improve the mobility and reduce the freezing. And all groups showed increase in exploration in the top zone entry, because when the zebrafish is feeling comfortable, they will explore more on the top zone. And when the fish under a lot of stress and under a lot of negative condition, they will suffer and then they will prefer to stay in the bottom. So with the treatment, you can see the fish is now feeling comfortable, more comfortable, and then they start to enter a top zone and explore in the top zone more compared to when the fish is under stress. And with the entropy decrease only in BDLLA group. And for the conclusion, BDLLA is maybe have a little bit of alter effect to the zebrafish behavior, but in the combination, which is the optimal, and combined with the PLLA. So PDLLA and PDLLA combination with PLLA support the behavior recovery, and through that proof that the biocompatible of the biomaterial. To investigate how PDLLA and PLLA combination enhance the wound healing, QRT-PCR was used to assess the immune and inflammatory gene expression. So there is some conclusion we can extract from the figure, like CD109 was significantly upregulated in the treated group, and this suggesting the improved endothelial activation that may help the tissue repair. And in the both wounding group, with and without the treatment, had elevated in CD79 genes, this suggesting the B cell involvement. And interestingly, YL2A and CD3E were significantly higher in the polymer treated group, this reflecting an improvement in T cell coordination. And why the NOS2B was upregulated in both group, ARG1 was higher in the treated group, this indicating a shift toward anti-inflammatory macrophage activities. Additionally, BRKDC was down-regulated in the treated group, this implying the reduced excessive inflammatory. And with all of these results suggest that BDLA plus PLLA combination treatment promote a balanced immune response that help support efficient healing without overstimulating inflammation. And to explore the mechanism between the beneficial effect of the BDLLA and PLLA on zebrafish wound healing, QRT-PCR also was performed to measure key gene expressions, and this including 12 genes that involved in the wound healing process. So we can extract from conclusion based on this graph. So why the most genes show similar expression across the group. Uh, Notable differences emerge, like the team 2B was downregulated in the wounded group, suggesting the control matrix degradation. In addition, collagen 1A2 expression in PDLLA and PLLA treated group was comparable to the control group, where the untreated group displayed a statistically higher expression level of the genes, this indicating a stable extracellular matrix formation. And similar trends were seen in other collagen genes, but without the statistically significant. And importantly, VEGF expression was higher in the polymer treated group, indicating in angiogenesis. This is very crucial for nutrients delivery and regeneration tissue for the wound healing process. So this gene expression change support the role of PDLA and plus PLA in promoting efficient wound repair. This is the summarize of the biomarker expression and cellular response in the zebrafish skin wound after receiving the PDLLA and PLLA treatment by oral administration. So after the biomaterial are delivered into the zebrafish by oral route, now they already in the semestic system and they start to degrade. By degrading, they release some bioactive factor that can help with the ECM remodeling, angiogenesis, and immune regulation that involve in inflammation and macrophage polarization, and all of this will contribute to enhance the wound healing outcome. For the conclusion of this study marked a meaningful step forward in the wound healing research by integrating innovative biomaterial with precise lab-developed laser-based skin ablation method used in zebrafish. We aim to access the therapeutic potential of the PDLLA and PLLA biomaterial. This is commonly used in aesthetics application, and by repurposing this biomaterial for medical application, we explore their ability to accelerate wound healing with biosafety. And by building our previous developed laser technique, which will provide reproducible and consistent wound using the precision motorized control, we establish an ideal model for real-time wound healing study. And this platform allows us to investigate the biological effect of the polymer-based treatment under tightly regulated. And by focusing on immune regulation, extracellular matrix remodeling, and angiogenesis, we conclude that PDLLA and PLLA treatment can actively enhance skin wound healing. And for the conclusion, our platform can be used to test potential biomaterial, therapeutic compound, or drug delivery system to enhance the wound healing outcome. That's all of my presentation. Thank you for listening.